From Microbe TV, this is Office Hours for Wednesday, January 17, 2024. I'm Vincent Racaniello, and joining me tonight from Ithaca, New York, Cindy Leifer.
Hi, everybody. Welcome. It's great to be here. I think this is so exciting. I've been watching these, and I was like, when do I get my chance? So now's my chance.
Let's welcome everyone. Thank you all for coming tonight. The moderators are here as usual. We have Les, we have Andrew. So Les is in the Bay Area. Andrew is in New Zealand. We have Steph SF, San Francisco. Tom Steinberg is in Eugene, Oregon tonight. And I believe that does it so far. Maybe others will show up. And thank all of you for joining us tonight. I hope you got your immunology questions queued up. Here's your opportunity to find out if COVID vaccines make you magnetic or not. No, just kidding. Let's find out.
That's an easy one.
From we have Noir from Santa Fe. Welcome, Noir. Gallbladder 8 is in Baton Rouge. Joseph is in Southern Ontario. Rima is in Iowa. Visto is in Australia. That's our farthest so far. I think Australia is the farthest possible from us. That's what Andrew said.
Where it's never snowing. See how he says where the day where it's never snowing. It's snowing here in Ithaca, so we have enough for you.
The New York weather lately has been awful. It's freezing. Yesterday it rained, and now all the rain is frozen. The cars are covered with a sheet of ice. It's treacherous to walk around. Oh my gosh. Even in New York City, there's ice everywhere. And it's still cold. John is from Minnesota, 8 degrees F. Wow.
Oh. Yep, that's about what it is here. So right there with you.
I know Abdulaziz is from Saudi Arabia. That's pretty far also. Kang is in Chicago, where I'm sure it's cold. MK is in eastern Massachusetts. Nicola is from Italy.
Ooh.
Coralie is from New South Wales. Richard is in Portland, Oregon. Questel is in Columbus, or somewhere in Ohio. I know that because she has been here for every office hours since the first one.
Cool.
Simon is in the Bay Area. Major echo. Yeah, I'm sorry if there's a major echo.
Oh no.
You don't hear a major echo, do you?
I don't hear any echo from you or me.
Who's the echo on? Cindy is echoing. Okay. Do you have?
I'm echoing.
I don't hear it on you. Do you have? The sound is coming out of a speaker. Is that right?
In my earbuds.
Oh no, you shouldn't have any echo there. Maybe it's on my end. I don't know, folks. I'm unfortunately using a new deck, and I did test it yesterday.
I'm just going to check my settings.
And it was fine.
Check sound.
Do you have any echo control on your end there?
No. Microphone. Turn it down.
Let's look it up. How do I reduce guest echo in Ecamm Live?
Play sound.
So there's a sound levels. There's a thing called echo cancellation. Do you see it there?
Where is that? In.
So there's a. Well, maybe it's mine. Let me see.
Oh.
Sound levels. Where is it? I don't see any echo cancellation. I mean, I do have you here. Checkbox in the sound levels window. There isn't anything there. Nope. All right, someone's texting me about this. Let's take a look at that.
Everybody's saying, Cindy is echoing.
Yeah, I know.
Oh, no.
I don't know why. I know Cindy's echoing, but no,
You're all putting it in the chat. We get it. We're trying to figure it out.
Let's see here. So you don't find anything on your end.
Hold on. I'm looking here. Hold on. Is the settings? Webstream cam, microphone. Nope. I don't have any settings that I can change. My microphone is set. Oh, no. This is not good, guys. Huh.
It says when you wear headphones, echo cancellation is disabled. Now, I have a setting for echo cancellation here, but this is for me, not you, so I don't think that's going to do anything.
Could try not using the mic and headphones, but that might be worse. I don't know.
No, I mean, it should be... it should be 0 with headphones. You got... And there's no sound coming out of your computer. You're pretty sure about that, so you don't hear me.
Yeah, nothing coming out of my computer. It's all my earbuds. Huh.
All right, folks, tell me. All right, you said major echoing. I got that. How major? Is it something you can handle? She has no speakers. She's just got earbuds. Right? We just went through that. So that's not it.
No.
Let's see if I can.
Let's try one thing. All right.
I mean, I don't think it's my thing here.
Let me just try this. Maybe it's still the same mic, but I chose it a different way.
It's a delayed copy of the same signal being mixed in on Vincent's end. Okay, so let me try a couple of things here. So do you still hear me, Cindy?
I do.
All right, folks, I'm turning down all the external outputs here. And you tell me now if that solves the problem. There's not a mic on the earphones. So I just... Yeah,
no, these earphones do not have a mic.
And
I'm using this mic.
It's not Cindy. I have it's not really tolerable. Okay, so I will try and fix it here. And if we can't fix it, we're going to have to stop.
Oh, no.
Which is unfortunate. It's not really tolerable, the echo.
All right, let's try. Let me see if I can just...
Immunology question. Oh, wait, you have to wait. So someone tell me, it's gone. Vanity Nutrition said it's gone. So
wait, gone?
Whatever I did, I turned off all the other sources. Excuse me. And that takes care of it. All right, Cindy, we're good.
Okay.
I have some skills, apparently.
Oh, yay. Okay.
Oh, now I can't hear Cindy. Wait.
Oh, no, you can't hear me?
Let's see. Cindy, can you count to 10?
1, 2, 3.
Hold on. Keep going.
4, 5, 6, 7, 8, 9, 10.
Is this still
They're
gone,
all saying
folks?
it's fixed, but can you hear me?
I can hear you now because I turned up you on a separate channel, and
Okay.
I think I did it. No, I didn't turn it on and off. So I have a new board here, which is this big as opposed to this big, and it's back, huh? Are you sure, Noir, it's back?
Yeah, but did you see this? That's what we used to call the NASA fix.
No. Wait, so the echo is right here on this. Cindy, keep counting again. I got to figure out how to...
1, 2, 3, 4, 5. Maybe I'll say immune cells. Macrophage.
Why is it mixing
T
her
cell,
back
B cell,
into the mix?
eosinophil, basophil. You guys
she's
are
going to
really
be back
patient.
because, so I have to turn this volume up in order to hear her.
Oh no. But if you turn it up, then it's back.
No, if I turn the volume up so I can hear you, then it comes back. So.
Oh no.
Wait, we're seeing still good, gone. Let's just keep going.
It says I'm good. Can you still hear me, Vincent?
Yeah, I do. So it doesn't make any sense that.
Okay. Well, everybody's like echo's gone, so let's just go with it and hope that we're good.
Okay, where was I? I was looking at where people were.
Now we need a timestamp. Ignore all the counting and technical issues. We did log on early and we did not hear anything, so we didn't think it was a problem.
Yeah.
Sorry.
All right, let's go back to seeing where people are from. We got David from Dayton, Ohio. We have good questions here. We have Elicio is from Brazil. Will is from China, Changsha, China. Cheryl is from Santa Rosa. Jessica is from the University of Toledo in Ohio. Okay, it's still good now. Fields of Green is from Salinas. LRD LDY is from Frederick, Maryland. Pete is in London, UK. Anatomy Geek is in Vancouver, BC. Anatomy Geek, yeah. Echo. Now I got more echoing here.
Oh no.
Excuse me. But no, these are old.
Okay. You're just looking through the thing because I was like, there was
Barry
lots
Ozone
of questions
is from Asheville, North Carolina. Hat is from LA. Sam is from snowy Western New York. Elmugger 1 is from Pueblo, Colorado. Oh, look who's here. Amy Rosenfeld, welcome.
Hey.
How's the sound, Amy? Is there any echo? Tallahassee, Florida. Oslo, Norway.
Ooh, that's a fun one.
Someone else from Brazil, Jacques from Brazil. From Singapore, who's got questions. We'll wait to get those. I should stretch these questions across both.
Oh, okay. Because my poor little eyes, I said I can't read that.
Yeah. What I did was cool, but I don't want to talk about it. It's has nothing to do with earbuds or mics or anything. It's having to do with subtracting it from my mix. But it's working. Perfect. It went away. Okay, I think we got everyone and their locations. This is primarily for Cindy to show how far and wide. Artemis is from Montreal. Wayne is from Urbana, Illinois. Steve is from Indiana. Peter is from Winona, Minnesota. Sharp is from Vancouver, BC. Charles is from Lyndhurst, Ohio. MN is from near DC. Vancouver. All right. That should do it. Let's go to the top and take. So, Cindy, why don't you give us a brief history of yourself? Let me see. You were born in Baltimore. Is that right?
I was. Yeah, I was born in Baltimore. And I went to my undergraduate at the University of Maryland, College Park. And from there, I moved to New York City, where I went to Weill Graduate School, Medical School, Graduate Sciences, or whatever. I forgot exactly what they call it, where I did my PhD. And that's where I fell in love with immunology, but I also got really interested in innate immunology and how our immune system recognizes infections and how we can use that to gain information to be able to maybe make vaccines better or immunotherapies, or think about blocking those kinds of responses when they're overactive in things like autoimmunity. So that's where I got interested in that. And so I loved living in New York City for the time that I was there. But then I decided to do my postdoctoral fellowship at the National Institutes of Health in Bethesda. So there was somebody from DC here, so right close by there. And the NIH is just a absolutely fabulous place to do science. It was super fun being there while I was there. And while I was there, I worked really on, again, innate immunity, sort of the microbial patterns and structures that our immune system recognizes and how they recognize them and how they signal. And that I got really interested in that. And from there, I moved up north to Ithaca, New York, where I started a faculty position at Cornell University, and I have been here.
You're breaking up, Cindy.
Uh-oh.
Oh, now you're good. Okay.
This is
so you're at Cornell.
bad.
You've been there a long time and doing immunology
Yep.
research. When did you join Immune?
Oh, have we been doing this for 6 years now? In 2018, I think, is when we started. Does that sound right?
Yeah. We're on
I think it was
episode
2018.
70 something, right?
Yep. Yeah.
All right. Let's
And
take
do they
some-
know that? Do they know the story of that? That's
no,
a
why
fun
don't
story.
You could tell them.
Yeah.
You could tell them.
Yeah. So I had known of Twiv and Vincent, and in science, I don't know how many of you know this, but we all go around and travel and give talks at other institutions. And so we had invited Vincent to come and give a scientific talk. And he gave an absolutely fabulous scientific talk. And in the last 15 minutes, he talked about science communication. And when we have faculty visit us, we have an opportunity to meet one-on-one about them and tell them about our research and, just like socialize and get to know each other. And Vincent was in my office, and we were talking science. And right before he walked out, I said, I really like your podcast and whatever, but when are you going to do an immunology one? He goes, well, as soon as I find somebody who'll do it, will you do it? And I said, oh gosh, I don't have time for that. There's no way I have time for that. And so he was like, okay, well, if you change your mind, let me know. So he walked out, but the seed was planted. And I thought about it. I was like, oh, I really want to do that. So I emailed him back and I was like, I think I do want to do that. He's okay. I've got a third person. Let's do it. So he already knew Steph Langle was interested in that. And then I guess it was in 2020 that we brought Brianne on as well. And so now it's the team. Yeah.
Yeah, without you, it wouldn't have gotten started. So thank you very much for joining and for your continued enthusiasm. It's really good.
Yeah, I love it.
All right, we have a question from Kang, and maybe...
Okay.
Yeah, this is probably something that has to do with CTLs, but you could probably answer it. Early in the infection, before viral proteins make it to the cell membrane, are there signals that allow CTLs to detect a virus-infected cell?
So CTLs are cytotoxic T lymphocytes, and they are going to interact with very unique, I call it like a fingerprint. It's a sequence of amino acids that gets presented in something called MHC. And that's what they recognize. So without that, they're not going to be able to know that the cell is infected with a virus. That's how the infected cell tells the cytotoxic T lymphocyte it's infected. It puts that chunk of the virus in this little MHC, and then the cytotoxic T cell comes along and recognizes that and kills it. So I don't think that before that those peptides get presented that the CTL has any way to recognize that infected cell. They can hide, but there's innate mechanisms to recognize that will help jumpstart the immune response.
Okay. We have a contribution from Kip and Laura. Thank you so much for your support of science communication. Hi, Vincent and Cindy. Parents want their child to have as little contact with the med system as possible. Mm-hmm. One way of keeping him, her out of that system is to take advantage of vaccines, a highly effective preventative medicine.
Absolutely. 100% agree. That is the best way to keep a child healthy is to get them vaccinated against all the things that you can protect them from. No doubt.
All right. HAT writes, patient had mono EBV. I don't know what S/P is. Do you know? For 10 years. Every time receives a vaccine or has a new infection, viral, experiences dizziness and shortness of breath. Common syndrome or just individual reaction? MD sees nothing.
Well, I am not 100% sure of this. EBV is a chronic viral infection, so it's always going to be with you. It's a DNA virus. Sometimes inflammatory stimuli can reactivate them. I don't know if you know more about that, Vincent, than I do. So, getting a vaccine might trigger some inflammation, but I don't think there would be a reactivation of EBV all the time with that. So it could just be that they have some reaction to the vaccine. Some people do have some slight reactions. It's usually injection site. It's not usually dizziness and shortness of breath. That's a little unusual. But those dizziness and shortness of breath also happens when people are scared of needles. So if people get anxious around getting a vaccine and they hold their breath or things, that could also contribute to it as well.
Yeah. All right. Simon wants to know the latest on long COVID and what immune problems may be involved.
Yeah. So I did look into this because I figured we'd get a question on this. I know that there was a very recent paper within the last week or so that came out of a group at Penn, and they've done really extensive characterization, and they found some problems with T cells that in the long COVID patients, they are a little bit more what we call exhausted. So basically they just are overactivated and they become a little bit refractory. And the other thing they noted was that there was some delay in antibodies production from I can't remember whether it was, I think it was the long COVID patients, but there is definitely some more data coming out now that we're beginning to understand a little bit more granularity in the molecular mechanisms of what's going on. But some of the things that I'm reading is really suggests that maybe there is, and maybe you can comment on this, Vincent, is there might be a reservoir where there's still some replicating virus because the T cells being exhausted sort of suggests that they're continuing to see antigen. I don't know if we really know if that's in fact true or where that might be, but we're definitely learning some really interesting things.
Yeah, some people think there is a reservoir of replicating virus, but I don't see. We need to get some evidence for it, right? And we don't have that.
We do. We don't have that yet. We just have a signature of T cells that look like they're still seeing antigen.
I mean, there was that paper. We did this on TWIV a while ago from Akiko that looked at a lot of immune signatures, and one of them was actually EBV reactivation, right? And was the other one some kind of macrophage? I don't even remember what it was. Do you remember that?
No.
Okay. But then other groups find other things, so it's also a matter of finding some commonality, which is difficult.
Yeah.
So 2 people have said that the EBV patient sounds like a vasovagal anxiety reaction. There you go.
Yep.
Which is what you said. Okay, Rima has a question. The eye, is there not immune cells that carpool there? Immune cells hardly ever get into the eye?
Oh, yeah. I'm not an ocular immunologist, but we usually say this is what we call an immunoprivileged site. And the idea there is not a lot of immune cells that get in there. And if you think about it, it makes sense. You don't really want immune cells going in and destroying tissue in the eye. That is not, you need vision. Well, 100% need vision, but it's really helpful. So immune responses in the eye are very well regulated. And if you think about the lung is not a great example because we still get really robust immune reactions there. But basically, in a lot of the tissues where its life is dependent on the function of the tissue, the immune responses there are very highly regulated. So it's not that you can't have an immune response in the eye. Certainly, if you have a trauma to the eye, then you can lose vision because the immune system can what we call break tolerance and attack the tissue in the eye, and then that will be destroyed. Yeah, I don't know if that was a great answer for that, but
a number of years ago, Ebola patient who had recovered months later was found to have Ebola virus in the eye.
oh yeah, in the vitreous fluid in the eye. Yeah. His eye turned color. I forgot his name, but I saw him give a talk. So it was really cool. So he was in one of those like doctors borders or whatever there, that he was a physician, an infectious disease physician, and he was treating all these patients, and they had to measure, take their temperature twice a day. And between the morning and the evening, he ended up catching it. And he doesn't remember anything like an hour or two after that. But apparently they were collecting all of the samples and fluid and everything. And they did all these treatments for him and monoclonal antibodies and all this kind of stuff. And the talk he gave was actually the data on that they had collected from him. And he went through, like, oh, here is the first sample they collected. And here, this, and this happened to my T cells and my antibodies and this, that, the other. And then he's like, and this is where I became conscious again. And then, these are my immune parameters. It was really fascinating to have that. But yeah, a year later or whatever, he still had Ebola virus in his eye. It's like crazy.
His name is Ian Crozier.
Yes. Yeah. He's a great storyteller too.
In fact,
what an experience.
He's agreed to come on TWIV. I just have to book it.
Oh, he'll be great.
He'll
be great. Yeah.
All right. John says, a previous office hours guest has said, quote, immunology is where intuition goes to die. What would be your first example of that comes to mind? It was actually Brianne who said that.
Brianne, no.
Right here on office hours.
Intuition goes to die. I don't know what she's talking about.
So is it really the fact that the immune system doesn't make any sense?
No, it makes a lot of sense. I think we just don't understand all of it. It's so non-linear. That's the thing that students have challenges to understand when we're teaching it, is that every cell talks to every other cell. I mean, we did a paper on immune with, Ann Kui. We were talking to her where, the symphony thing. So analyzing the response of lots of different cells to lots of different cytokines. So each cell can respond differently to the same cytokine. And each cell secretes many cytokines, and then every cell can respond to many different cytokines. So it's so complex and interconnected. I think we just don't understand it yet. It's not that it doesn't make sense. It makes sense to the system. It's just we haven't probed the system enough to understand it yet.
All right. Here's not an immunology question, but interesting nonetheless. Are there any studies on infection frequency and personality type? Are introverts less likely to be infected because they're less likely to socialize?
I think you answered your question. I think introverts who sit home and read a book are going to be less exposed to infectious agents because they're not going out to the bar and breathing all the contaminated air.
They have to go to the grocery store at some point, right?
They do. Well, no, they probably are more likely to order their groceries online than an extrovert. The extroverts I know go to the grocery store because that's where they want to meet everybody and socialize.
I don't know of any studies that look at it. This would be a hard study to do, right?
It would, yeah.
For many reasons.
Yeah, because you don't know where someone's going to get exposed, so you can't really track it back very easily.
So Hat says, it's okay. I usually have to listen to Immune twice to understand it anyway. Oh, boy.
We try to explain.
Well,
catch us,
that's a goal, right? To try
us.
And make it clearer, right?
Yeah.
All right, so our friend from Singapore, whose characters I can't read, says, I have two questions for Cindy. First, does vitamin C play a significant role in human immunology?
I would say yes. I don't know any specifics of that, but if I had to guess, pretty much everything plays a role in immunology. I can tell you vitamin D, we know for sure. There are definite roles for vitamin D in generating regulatory immune responses. But vitamin C, I don't know. They always tell you to drink your orange juice, right? Because it helps your immune system. But lots of immune cells have requirements for, vitamins and other things in order to perform their best to protect us. So my guess would be yes, but I don't know the specifics of that one.
Let's see. The second one is how do subunit vaccines like Novavax generate good CTL responses?
They don't normally generate very good CTL responses, but how they would generate CTL responses at all is because we come back to what I was mentioning at the beginning with these, the virally infected cells put the peptides in the MHC. Well, the way this whole thing gets started are what we call antigen presenting cells. So they go around and they take up bits of different, pathogens, and they put them in these MHC molecules and they activate the T cells. And that's how the T cell response gets going. So some of these dendritic cells or antigen presenting cells, that's the detailed version of it, they can take up the antigen and get it into the right MHC in order to activate CTLs. Usually they would put it in to activate what we call helper T cells, there's two subsets of T cells, CTL or cytotoxic T cells and helper T cells. So normally if you take up an antigen, it'll activate a helper T cell, but they can also activate CTLs. So, but the bottom line is very inefficient. Most subunit vaccines are going to generate really strong antibody responses and not very good CTL responses.
All right, here's a polio question. What are the immunological difference between a person who got OPV and a person who got IPV at the time point years after vaccination? For example, is gut IgA still different after years?
So this comes back to the last question where it was a subunit vaccine that does not induce very good T cell responses. If you have an oral polio vaccine, that's going to be generating an immune response at the mucosal surface, so in your mouth, and then that will generate those same kind of immune responses at the other oral surfaces, so your nasal passages, your gut, and that's what's going to help protect you from the polio virus, which is oral virus, right? So yes, IgAs are a major part of that. And IPV goes in the muscle, and that does not generate a very good IgA response at all. It's typically what we call an IgG response. So that's going to be in the blood and not at the mucosal surfaces. So there is a difference between the immune response that's generated from OPV and IPV. And that's why OPV is more protective in general. However, it has its limitations, right? So every time we think about a vaccine, we need to think about the cost and the benefit. So it's a very good mucosal vaccine, so it protects the right place at the right time, but it has, some potential to revert and whatever. So IPV is safer, but doesn't generate the right kind of immune response, which is good enough if there's not a lot of the virus circulating in a population. So that's when you can switch over to IPV because it is protective enough if there is very little exposure. Do you have any tweaks to that?
So yes, I wanted to say that, yes, you're right. The OPV does induce mucosal IgA, although it does drop very quickly after immunization. And so people who have OPV, say a year later or 6 months later, can still be infected and shed some virus. But in both cases, you have protection against neurological disease, which is really what the vaccine is doing.
The important one, right?
Yep. Dagfin has a question. There are a few recent studies pointing to reactivation of EBV or HHV6 in COVID-19 patients. Do you think this could be a causative relation by a specific mechanism, or are we seeing statistical noise manifested as increased incidence of reactivated EBV in COVID-19 patients?
I'm not 100% sure, but if I had to take a guess, I would say, when you get COVID-19, it's causing some loss of immune responses become a little bit immunosuppressed, allowing the virus to reactivate.
Yeah, I think also there is the stress of having COVID, the inflammatory response probably contributes to reactivating EBV as well. So I think it's a specific result, not just statistical noise, right? Are you there, Cindy? Cindy's frozen again. So Cindy periodically freezes up. Okay. Let's see if she'll unfreeze. Yeah, it's very cold up there. Now she's gone. Oh, dear. Well, I can amuse you easily until she comes back. I'm sure she will come back. And so there's some, I'll have to mark these questions and come back to them because they're immunology questions. But I can handle this one. How does measles wipe out immunity to other prior infections? Oh, here we go. I'm going to answer this because she's here.
All right.
Hello.
Sorry. I'm back. I should have done this from my office.
It's okay. We're getting a lot of chat in. Is your son gaming or something?
I don't think so. Oh no, now we're saying echo again. Shoot.
Really? Echo again?
I have all the settings the way it was. Okay.
Let me see. All right, let's get a response. Is Cindy echoing still?
Check. 1, 2, 3. Macrophage. Am I echoing?
Cindy has echo. No echo on my end. No echo. Okay. We got no echo, so that's good. All right, here's a question.
Okay.
Here's from John. How can you give me a reminder on IgA, IgG, Ig? How can a layman keep all those straight? Or IgG the good ones?
They're all good. Okay, so they can all be good and they can all be bad. It just depends. So IgA is the one that's going to protect your mucosal surfaces. So that's a really good one to have, for example, against flu or COVID-19 or whatever. IgE, we always say it's bad because that one is associated with allergy. So if you have hay fever, if you get like allergic to cats, all of that is going to be driven by IgE. However, IgE is good against parasites. So although hopefully you don't have intestinal parasites, that's really good against parasites. And then IgG is the main one that we typically study because that's going to be the highest concentration in the blood. And so that's really good at binding up some viruses and bacteria.
Okay. All right. MK wants to know, how does measles infection wipe out immunity to other prior infections? Is it permanent?
Oh, so there was a paper, and I wish I could remember the details. If I had known this was happening, I would look that back up. And I'm pretty sure,
Mina, remember?
Yes, it was Mina, and we covered it on Immune, right. I think. And it has something to do with, it damages the memory cells and it wipes them out. So yes, it's permanent. It's not, you're not saying like everybody who gets measles is going to lose all their memory cells. That's not the case, but they did show pretty convincingly that when you have a measles infection, you're going to mess up the memory cells. And therefore, if they're gone, they're gone. You can generate new ones by getting immunized again, is basically how you could get around that.
Or infected, right?
That would better to get immunized than infected. I recommend vaccination versus infection. Yeah.
All right. Hannah wants to know, if you had to teach immunology to undergraduates in 3 lectures, what topics and concepts would you emphasize?
Spur of the moment, I would say you need to talk about 1 lecture on innate immunity. So how we recognize and respond to infection, because that's my area of interest. So I got to do that. The second lecture would really be, I think, on, I would use a vaccine as an example and go through, like, when you give a vaccine, how you generate T cells and B cells, and what they do to protect you. And then the third one, I think I would really get students excited by talking more about different immunotherapies. So what have we been able to exploit, what we've learned about the immune system to create therapies to help people? That's what I, in a very broad way, that's what I would do in 3 lectures.
Wow. 3 lectures on immunology. That would be tough. All right. Pete says, Cindy, what's the history of the nomenclature of T cell names like CD4, CD8? Why CD? Are there other important CDs?
Yes. Okay. So CD is cluster of differentiation. We never actually say that out, spell that out, but it's cluster of differentiation. And the history of that is that people made antibodies to things. And then if a group of antibodies recognized the same thing, that was a cluster. So if there were a cluster of antibodies that recognized a T cell, that was a cluster that recognized that T cell. And then you could start parsing out, this antibody separated that T cell cluster into 2 different clusters. So then I get a little murky exactly how they did all of that, but that's generally what they did. And so it's a cluster of differentiation. And so 4 and 8, they were able to differentiate 2 types by different sets of antibodies that recognize one set of T cells versus another set of T cells. And then they were able to say that the CD4, this cluster, was a helper T cell, and this other cluster was a killer T cell. And so that's how they got to that.
Even the molecule that my lab discovered, the polio receptor, has a CD number, CD155.
Yep. Yeah, so basically every surface molecule now has a CD number.
All right, Carol writes, my platelets dropped after my second COVID vaccine, then back up. They dropped again after the third, then back up. Is this immune mediated?
Probably, yeah. I don't know what would cause that.
Yeah, I don't know either.
There's a lot of connections between the coagulation system and the immune system. So inflammatory stimuli can cause hyper or hypocoagulation. So there could be things related to platelets in there too. But yeah, I don't know why it would cause a drop in platelets.
All right. Susan writes, can you explain what components of the immune system innate causes the feeling of malaise? If we knew, we could begin to understand post-viral syndromes like long COVID. We need your brain.
So this gets into something that I'm kind of interested in. I was just talking to Vincent before this about what kinds of things we could do if we do a second immune per month and what we might talk about. And so some of the things we might talk about would be different cytokines and what they do. And the reason why I started with that is because a lot of the innate immune responses are driving particular cytokines. So we always group together these 3 cytokines we call IL-1, IL-6, and TNF. And those have various different effects on the immune system. Some of them including they get into the brain and they act on the hypothalamus to raise the body temperature. And so getting a fever makes you feel pretty crappy. But in general, these cytokines are very broadly active. They upregulate, they stimulate the liver to upregulate things we call acute phase reactant proteins. And they just generally activate a lot of different cells. And that is going to contribute to it. So if very levels can contribute, and they can also contribute to things we call cytokine storm in different immunotherapies. And those all contribute to this malaise. That's going to be a contributor to a major part of this. Also, some of it might be just some immunosuppression if your immune system isn't working well, or you have different kinds of cytokines or things being produced that could contribute to it. And I don't know how much of it is studied, but there's probably some muscle aspects to this as too. So chronic inflammation may have effects on muscle, and you feel weakness and tiredness.
Lise wants to know how you became interested in immunology.
So in college, I took a whole bunch. I thought I was going to be a physician, but organic chemistry beat that right out of me. But then I took biochemistry, and I loved biochemistry, and I was like, okay, wait, I like this. And I took some cell biology and microbiology. I really loved those. In my senior year of college, I liked immunology better, so I went into immunology. Yeah. And then once I was in graduate school, I just loved immunology.
Okay, Sam writes, does repeated COVID infections disable or harm immune systems, leading to reduced ability to cope with illness and systemic issues?
Well, your body's really good at fighting infections, especially infections you might have frequently. We get colds every year before we had COVID, right? A lot of people would get sick every year. And it doesn't necessarily mean that your immune system is disabled per se, but we do know that COVID is not great for your immune system. So in the short term, it might cause a little bit of immunosuppression and make you more susceptible to other things. I think that you should generally recover from that unless you have long COVID. And so it wouldn't cause like long-term systemic issues yet that we know. Doesn't mean that we won't find that out later.
So we have 218 folks listening in. That's great. And only 94 likes. Folks, hit the like button. It's right below the video there. That makes us more visible to other people. Thanks for that. Doesn't cost you anything.
Yeah.
All right. Visto says, how do immune cells know what is self when experimenting with immune targets?
Yeah. This is great. This is like what I really love about immunology and the innate immune system. So for T cells and B cells, it's easy. Okay. They are recognizing a structure, and they don't know where that structure comes from. That could be self, it could be an antigen in your toe, or it could be plague. They have no idea. And it's the innate immune system that provides that context to know whether it's self or non-self. And the way they do that is through the receptors that I study, which are called innate immune receptors. And they tend to recognize microbial structures generally and trigger the immune response. So they initiate the immune response, and then they also help provide that context for the T cells and B cells when they're going to respond. So if a T cell is responding to an antigen, the context from the innate immune cell will be telling it, yeah, that's foreign, you should respond. So generally, it's through specific receptors that will recognize things as foreign through the innate immune cells. That's why they're pretty cool.
All right. Gallbladder 8 says, regarding germinal centers and lymph nodes, might there be advantages or disadvantages of receiving a vaccine injection in the same or opposite deltoid to tickle previously tickled germinal centers?
So we have covered this on immune, and we had Gabriel Victora on, and he talked about memory generation. And we also had someone else on that talked about that too. And maybe you'll remember the name or look it up. But from what I'm hearing, yeah, I always get the injection in the same arm because that way your germinal centers are already there, and that's the draining lymph node to that arm. And so you will reboost those immune cells. And I think there's some data out there that says that is better, that you generate better immune responses if you go in the same arm.
Didn't Gabriel Victora have some on that as well?
That's what I thought, yeah.
Yeah. Okay, Mike and Lori write, could you explain the role of CD8 killer T cells in autoimmune disorders like oral lichen planus?
Okay, I don't know oral lichen planus specifically, but CD8 killer T cells that are involved in autoimmune disorders are typically autoreactive. That means they're recognizing a self antigen, and if those healthy cells of your body have that antigen, those CD8 T cells will kill them. The most common example that I know of, so it's not your example of oral lichen planus, but it's type 1 diabetes, not type 2, but type 1 diabetes. It's an autoimmune disorder where the individual develops CD8 T cells that attack the beta islet cells and destroy the beta islet cells. And so the beta islet cells are the ones that make insulin, and those individuals become insulin dependent. So they have to take insulin to supplement their own body because they can no longer make the insulin because their immune system attacked and killed their beta islet cells. So I don't know oral lichen planus, but probably if it's a CD8 T cell mediated autoimmune disorder, it's going to be the same type of thing where there's some sort of autoantigen. Now you could ask me, how does that happen? I just told you that the CD8 T cells get context from the innate immune system so that they only will attack foreign things and not self things. But there are times where we don't really understand exactly how it happens, but some individuals will break tolerance and generate an immune response to self antigens and attack self.
John writes, which has more acronyms, virology or immunology? It's got to be immunology, right?
Probably immunology. Yeah, because we have cytokine, all the different interleukin cytokines and all the different CD molecules and stuff.
Yeah.
But yeah. And he asks about TLRs. That's my favorite. That's so TLRs. Yeah, so TLRs are called toll-like receptors, and there's a whole backstory to why they're called tolls that if you want, we could get into. But they are innate immune receptors that will recognize microbial structures directly. So the canonical one is gram-negative bacteria. So you might know they're gram-positive bacteria and gram-negative bacteria. Gram-negative bacteria have a molecule called lipopolysaccharide, and there's a TLR that recognizes lipopolysaccharide. So it will see any gram-negative bacteria will trigger that TLR. So it's kind of like a general, oh, there's a bacterial infection, there's a gram-negative bacteria, you better induce a response. So that's what they're there for, and they're poised on these innate immune cells and trigger the inflammation. And are there more than 11?
11, yeah.
Yes. So I think in some fish, there's like 25 or something like that. Sea urchins have something that's very similar, and I think they have like 100 and some. There's some really cool things about comparative immunology. But mice and humans have, between 10 and 13.
All right. Artemis writes, have you read the paper in JAMA, From Immunity Debt to Immunity Theft, How COVID-19 Might Be Tied to Recent Respiratory Disease Surges? And if so, what do you think of it?
I did not, but I want to write that one down.
I have not either, but yeah. I mean, this is the idea that somehow SARS-CoV-2 is specifically involved in the recent surges, not just, well, I guess immunity debt that's what it is. You haven't gotten infected with these other viruses for a while, right?
Yeah.
Let's see. Andrew writes, as a gardener, I am learning that sound can influence plant growth and health. I know that noise stress is harmful, but can it affect the immune system?
I'm trying to think if I've heard any example of this. I don't know. Lots of physical properties affect immune cells. So we've looked at stiffness. So tissues change in stiffness, and that affects immune cell function. Temperature affects immune cell function. Pressure, so like edema, when you get swelling and it puts pressure on cells, that can affect immune cells. So sound, vibration waves, maybe. I don't
know. David writes, how does the immune system react to endogenous retroviruses?
Well, in order for the immune system to see, quote unquote, see something, there has to be an antigen. endogenous retroviruses, do they produce antigens or do they just hide out in the genome, Vincent?
They can produce
Sometimes
antigens.
can.
Yeah, but they're there from development, right?
Yeah, so that is a thing. It's going to look like self.
Yeah.
So if there is an antigen and it's generated from birth, there is a process that each individual, all the cells in their immune system go through that generates the tolerance. So we get rid of all of the self-reactive, not all, most of the self-reactive immune cells, T cells and B cells to some extent during development. So if the retroviral protein is being produced from the genome, it would be just like a self antigen and tolerance would be generated against that. So probably it's not reacting very much to those endogenous retroviruses.
Okay, is severe viral disease primarily from immune
Mhm.
dysregulation? Would you compare how this works with two viruses in the news, COVID and severe measles?
So I think the important point to mention here is that pathology, so damage to the body during a viral infection, is from two different things. So one is the virus, depending on the type of virus and the cells that infect can actually cause damage. And then the other side of it is the immune system trying to eliminate that virus is going to cause damage. So either one could be happening, but I think a lot of the time it's from the immune response trying to eliminate the viral infection that actually causes a problem. And comparing COVID and measles, those are just very different diseases, but they're both, one is going to be in the lung and one is mostly in the tissues and in the brain. And they're just, you're gonna, I don't know how to answer this really. They're both going to be probably primarily the immune response to the virus that's going to cause a pathology. So in COVID, the immune response is causing the damage to the lungs. And in measles, when you're getting the rash and things, that's probably started by the virus causing tissue damage, but then the immune cells come in and trying to clear that up.
Okay, Dagfin asks, how is S protein from the mRNA vaccine released? Is it expressed on the cell surface or cleaved to soluble particles?
I think it's both. So one of my students studies a little bit about a feline coronavirus and also has a spike protein. And infected cells put the S protein on the surface, and they can the S protein can actually form what we call syncytia, so the cells will fuse to each other because the S protein's job is to fuse envelopes together, so to fuse membranes together, so to fuse the virus to the cell or fuse the cell to each other. So it can be on the surface. And if it's made in the mRNA vaccine, it could also get to the surface because the mRNA is going to go inside the cell and the cell is going to make the protein and some of that protein can go to the surface. But probably some of it is also released in particulates or shed from the surface of the infected cell. That can happen as well.
So the mRNA vaccine, the nanoparticles are going to be taken up primarily by APCs, right?
Yes. Mm-hmm.
And in those, it'll be made, the protein will be made intracellularly, then it'll be processed, I guess, right?
Yep.
And then the peptides will be loaded on the surface, right, for presentation to T cells.
Yep. Yep. So that's how we would activate the T cells. And to get the antibodies, it's probably a little bit of maybe some shedding and reuptake, or the protein gets internalized back from the surface and gets into endosomes because that's where it needs to be to activate the helper T cells in order to get antibodies.
Got it.
So there's two different, it's very complex. There's two different pathways that antigens go through in order to activate the two different types of T cells.
All right, Roberta says there are rare reports of mRNA vaccines inducing polymyalgia rheumatica. Any idea if Novavax has been implicated? The AstraZeneca adenovirus vaccine has been implicated.
I do not know if Novavax has been implicated. My guess would be probably not, because it's more of a traditional inactivated vaccine, similar to the influenza vaccine that we get from year to year, that doesn't seem to have those issues.
David asks, are there any studies showing that measles erases memory B and T cells for SARS-CoV-2?
I don't think it would be specific for certain memory B cells and T cells. I would think it would be able to eliminate those, just like they can for any of the other infections that they've tested. Yeah. So don't get measles, get the vaccine.
Yes. Frank says, any ideas on new ways one may tame a cytokine storm?
That's one of the holy grails, right? Because we get that in a lot of different things, in sepsis and in CAR T therapy and in a lot of other things. It's right now they keep targeting individual cytokines, IL-1 or TNF. Humira is one, anakinra targets IL-1, tezolizumab targets IL-6. But each time we target one cytokine, there's issues that happen, right? Because these cytokines have so many different activities in the immune system. So I don't know that we have good ways to stop it yet. Generalized immunosuppression, which also isn't great, right?
Forlos comments writes, I really don't agree with many of the papers claiming T-cell exhaustion in long COVID. There is a lack of functional assessment, cytokines in most, and in those that do it, it is similar to controls. That's a comment. You don't have to say anything if you don't want to.
Yeah, so I would check out I think it's Nature Immunology. John Weary's lab just published a paper. That was the one I was referring to. I think they did look at cytokines, but I could be wrong. I thought they did intracellular cytokine staining.
And also Forlos says it's not outside the realm of possibility, but that at least some of these long COVID patients have persistent infections driving exhaustion, but co-inhibitory marker is not equal to exhaustion.
Absolutely. The inhibitory markers that they use to label, to detect exhaustion, are also activation markers. So yeah, just because they upregulate, I'll use the nomenclature, PD-1 or whatever, it doesn't mean they're necessarily exhausted. You do have to test them functionally. Absolutely correct. Yep.
Anatomy Greek writes, how are epitopes selected to be placed in MHC and APCs?
Using all the lingo. Good job. So inside a cell, the cell is going to chop up an antigen or a protein into little bits. So that's going to be an epitope. And then that epitope is placed in the MHC. Now, how do you know which epitopes go into MHC? So there's we could talk for an hour about MHCs. They're really cool. Everybody has a different set of MHCs. Not exactly, but it's a very highly, what we call polymorphic protein. So there's lots of different ones in the population, and you have a selection of them. And what's different about them is they look like a hot dog in a bun if you look at the structure. So the hot dog sits in the bun, that's the peptide, but the bun comes up on either side. I'm showing fingers. And what's different is in the bottom of the bun, the amino acids that make up that bottom platform differ from MHC to MHC. And what that does is allow different sets of peptides to bind, because there's like little bumps in that platform that we call anchor residues. So that's little pockets for parts of the peptide to sit in. And so each MHC binds a different subset of peptides. So let's say one MHC will bind any peptide that has a leucine at position 2 and a tyrosine at position 8. I'm just, saying this. So any peptide that has those two will fit. And if you think there's 20 amino acids and the peptide is, let's say, 10 amino acids long, besides 2 and 8, all the other positions could have 1 of 20. So you can do the math. It's a huge number of peptides that combine to any one MHC. But they bind because they have these little, like, basically lock and key that fits into the MHC pocket.
Airy Ozone says, does having decades-long autoimmune disease affect how the immune system responds to viral or parasitic infections?
I think having any autoimmune disease definitely affects the response to new infections. And it can go 2 ways. So you're already hyped up, so you have a lot of inflammation. So your response could be over exaggerated to new infections, but at the same time, you can have a less robust and broad repertoire of cells to begin with to respond. So you could actually, although you're hyper-inflammatory, respond less well to new infections. So I think it just depends.
Any hypothesis why some vaccines give immunity for life and others only for a few years or months?
Well, some vaccines we had before. So, for example, the smallpox vaccine was a very, very strong vaccine. People had a lot of reaction to that. It was a live vaccine, and that we know can induce, we can still detect low-level antibodies in people 70 years later now, 50 years later. So certain vaccines where it's a very strong immune response, especially to like a live attenuated vaccine, will provide very long protection. I think part of it has to do with whether it's a whole pathogen or a chunk of pathogen. So recombinant vaccines don't last as long, but there's probably some nuance about the specific antigen itself and the length of time that it generates the B cells. Yeah.
So earlier we were talking about introverts and infections. So Nicola says, as an introvert, I can say lockdown was perfect for me. Yeah, that's good. Let's see here. I had another one. Oh, Craig, complement is the most confusing term I can remember.
You really don't want me to explain complement, do you?
But you agree
So,
it is rather confusing.
oh
Right?
yeah, it's I every time I teach it, I'm like, okay, we're going to talk about complement. It's complicated, and the students don't laugh, but I know they'll laugh later when they listen to it again. Yeah, it's a very, very important part of the immune response. It's at the ready. So all of the proteins are already made and it can act very quickly, but all of the pathways to generate it and its functions are really complicated. So yeah, it's one of those things that I didn't really like teaching for a long time, but now I kind of understand it after a long time and I can teach it.
Thank you, Abdul, for your contribution
Thanks.
to science communication. Oh, here's a question. Why do mRNA vaccines produce an unusually high fraction of IgG against spike? Of course.
I don't know why it's exactly IgG4, but I'm sure it has to do with the fact that the mRNA vaccines trigger TLRs, and it's the type of TLRs they trigger, and then the cells that they're inducing it on produce cytokines and conditions which preferentially generate IgG4. There's nothing wrong with IgG4. It's a good, they're all good immunoglobulins. It doesn't mean if you make a high IgG4 that you are immunocompromised or anything like that. Yeah.
So For Lost Comment says, I remember Professor Leifer's immunology lectures from my undergrad with great fondness. Undeniably among the best teachers and mentors I have ever had the honor of having. Okay,
Oh,
you probably don't know who
thank
this is, though, right?
you. I don't. I wish I knew who it was. I wish you put your name in there. Yeah, thank you. That's so sweet. I love teaching undergraduates. They are so confused at the beginning with so much nomenclature with the immunology, and then as things click and they come and they ask me these questions after class, and I'm just like, wow, you really get it, and I really have no idea how to answer your question. That's a fantastic question. So yeah, I love working with undergrads. Super bright and really excited.
Risto says, what is it with fever? Does it help reduce infections?
Yes, yes, it does. The immune system wouldn't do something as expensive as fever if it didn't work. So fever reduces the replication efficiency of both viruses and bacteria. They just don't survive as well with fever. And it also revs up the immune cells. So, not like super high fever that will give you, seizures and things, but elevated fever does rev up the immune response. The immune response can present antigen better, T cells work faster, B cells make more antibody. It's very interesting.
Forlos asks, what would you say are the major controversies surrounding the role of DAMPs?
Major controversies. Well, the one that I love is DAMPs. So first of all, we have to say what DAMPs are. So I think we talked a little bit about PAMPs, but I'm not sure we did exactly define it. So pathogen molecules we call pathogen associated molecular patterns. And they're the things that bind to those TLR molecules that I mentioned. So DAMPs are something called danger associated molecular patterns. Now, why do we define DAMPs versus PAMPs? Well, it turns out that DAMPs can be self molecules. And we went through this whole thing and described, self versus non-self, except that sometimes we need to recognize self. So, for example, a virus that doesn't cause that maybe paralyzes the ability of the immune system to put its peptides in the MHC, as an example. They still can cause tissue damage, and the immune system needs a way to see that. And so the immune system can sometimes see the damage that the organism made and not necessarily the organism itself. And that is one time where if cells are damaged and the guts of the cells are where they're not supposed to be, which is outside the cell, then the immune system can say, hey, wait a minute, that's not supposed to be there, and respond to it. And so we call those DAMPs. So those would be maybe cytoplasmic proteins or nuclear proteins that are supposed to be inside the cell. But if they're located outside the cell, then that signals danger. What are controversies around this? I don't know. I think it's pretty well established at this time that there are DAMPs and that they trigger TLRs and that that's required for inflammation. And you might say, well, when do you want to induce inflammation when there's not infection? Well, yeah, you can have autoimmune disease and that's not good. But sometimes maybe you bumped into a wall and you damaged some cells. You still have to have a way to recognize that tissue damage and repair it. And inflammation is important for that. There can also be like drug toxicities, where if you take a drug and it causes some liver damage or some kidney damage or something like that, then you have to recognize that damage and repair the tissue, and there's no actual infection around. So DAMPs are really important for that.
Lara writes, does a person contract shingles only if she had chickenpox before?
You can answer this one, right? Can you get a primary varicella infection and get shingles?
I don't
think
no.
so. I think it's always a reactivation.
Yeah. It's always a reactivation. Yep. Artemis says, do you have a favorite immunology book to recommend for beginners?
For beginners. For the immunology class that we start, we usually use a book called The Immune System by Parham. it's still quite detailed. You have to have a good background in cell biology and biochemistry to really dig deep and understand. But that's a good one if you have that background to start to learn immunology. I would also recommend that you listen to Brienne's lectures that she posts on YouTube. Those are fantastic. She's also an amazing educator.
Susan, thank you for your contribution to science communication. We really appreciate it. We can't do this without you. Nicola wants to know, what's the simplest organism that has something that can be called an immune system?
I want to say bacteria.
So you would call CRISPR an immune system and all the other defenses they have, yeah?
It is. Okay. It is. It's so CRISPR, that's how we found CRISPR, because it's a mechanism by which the bacteria can take little bits of phages, which are viruses that infect bacteria. They take bits and they put it in their genome and hold on to it, and that's memory. And it uses that to protect itself against infection from that same thing. So it's memory. It's really truly an immune system. So flies have innate immunity. Sea urchins have innate immunity. Yeah. Plants have immunity.
Sure. Okay. Forlos says, why would TLR9 be required for the development of anti-DNA antibodies in lupus? Apparently with TLR9 knockout mice, it doesn't happen.
Right. So the TLR9 stimulates the B cells to make the anti-DNA antibodies. And that's mostly how that happens. There's really interesting things. TLR9 is the DNA receptor, TLR7 is the RNA receptor. If you knock out TLR7, you knock out anti-RNA antibodies. If you knock out TLR9, you knock out anti-DNA antibodies. So it has to do with the immune complexes being taken up and stimulating the TLR inside the cell and then feeding back. So it's a B cell intrinsic phenomenon. But okay, so I don't know who you are. You're my student, but you should read some of the work that was done on this, because the wild thing is if you knock out TLR9, you actually make disease worse in mice, because the TLR7 is hyperactive. So there's this really cool interaction between TLR7 and TLR9. If you knock out TLR7, you knock out all the anti-DNA and RNA antibodies. But if you knock out 9, you knock out the DNA antibodies, but the RNA antibodies are up. And there's molecular mechanisms behind this. But yeah, it's very cool.
Sammy wants to know, if most COVID symptoms are due to the immune response, are patients on immunosuppressive therapies like TNF-alpha inhibitors at lower risk of severe COVID?
I don't know the answer to this, but immunosuppression is never a good idea when you have an infection.
That's the problem, Cindy. You're right, because they have no response early in the viral phase, which makes it worse. And so they never get to the inflammatory
so their
phase,
virus goes out of control and they can't
Yeah.
control the viral infection.
Yeah.
But that coming back to this actually is an interesting point, because when they were trying to do treatments with like anti-IL-6, so the tocilizumab, or steroids or whatever, you don't want to use them in the very beginning of the infection, because that will dampen the immune response and allow the virus to overgrow. But if you wait a little while till the immune system starts to clear the virus, and it's the immune system that's causing the damage, then you want to shut down the immune system. So that's when you would use cytokine inhibitory therapies or immunosuppressants or steroids.
Loretta wants to know if you predict mRNA vaccines are going to become commonplace for multiple diseases or medical issues.
Yes, I do, because they're easy to make. As soon as you have an antigen, in 3 days, you could start producing it, right? So if you want to generate a vaccine fast, then that's how you want to do it. And I think the other thing that we learned from this is that you get a better T cell response than you do from a lot of the traditional vaccines. So I think people are going to start moving towards this.
Kang wants to know if anti-idiotypic antibodies downregulate plasma cells.
That's a good question. So we need to say what anti-idiotypic antibodies are.
Right.
So those people with like lupus or other autoimmune diseases can have antibodies against their antibodies. So they can get antibodies. And those are called anti-idiotypic antibodies. So if you've got a lot of IgG and you start making anti-IgG, then you'll get antibodies that bind to your antibodies. I'm not sure that would downregulate plasma cells. So plasma cells are the B cells that are specialized antibody factories. So they can make milligrams of antibodies per day. And I don't know how they would feed back to shut that off necessarily. Yeah.
Sammy J writes, I'm currently in my intern year of medical training. I have been watching Microbe TV since 2020. Thank you for all the high quality knowledge. Thank you, Sammy. And I'm glad a medical doctor is listening because they think they need to.
Yeah.
Artemis writes, fever helps the immune system. Should we then avoid Tylenol and other fever-reducing medications when you have a low fever?
Yeah, this is something that we talk about all the time. And the question is, do you treat your fever or do you not treat your fever? I think if you have a low-grade fever and you can tolerate it, you should let it go. Because that'll give your immune system the best chance to try and fight it. But if you're really debilitated, and I mean, you get 104 fever and you are pretty darn sick, at that point, at some level, you want to try and lower that fever a little bit, because you run the risk of getting, too high a fever. And then you can actually have seizures and have brain damage. I know that when kids get really high fevers, sometimes you have to put them in an ice bath or at least in a cold bath to try and help lower their temperature and give them Tylenol. Yeah.
Ian, thank you for your contribution to science communication. Thanks. How far away are inverse vaccines to halt autoimmune pathology? We didn't do this paper on immune. We should probably. It's a cool paper.
We should. Right? Yeah. I don't know. Well, I can tell you that there's some cool ideas about using CAR T therapy to eliminate autoimmune B cells. But no, I don't know how close we are to inverse vaccines.
Hard to always say how close anything is to people, right?
Well, then, I mean, it used to be 20 years for a vaccine and now it's 11 months. So
apparently
things change.
Sammy J, who's our medical intern, I am always hesitant to give Tylenol for mild asymptomatic fevers in the hospital, and I always get looks from my colleagues. Like Dr. Leifer said, there's a benefit of fevers to an extent. Well, that's good to know. Les wants to know if you like Kyrgyzstan, the immune system cartoon animation series, yeah?
Yeah. My son brought that to my attention. So he is now a junior in high school. But a couple of years ago, he was like, oh, Mom, you gotta see this Kyrgyzstan. So he would show me the videos from it, mostly space stuff. He didn't show me the immune stuff, but I watched them. Yeah, they're really, really great.
Abdulaziz, who asked about IgG4, says, I know there's nothing wrong with it, but why IgG4?
I don't know. Yeah, I don't know. I don't know why. I think it's just really the types of responses that are generated. So those nanoparticles are going to drive certain types of inflammation, trigger certain TLRs, trigger certain cytokine profiles that will then just prompt the preferential generation of IgG4.
Steph says, Cindy, please, not an ice bath for kids. Evaporative cooling with wet towels or tepid bath, please.
Thank you. I remember being put in a bath and it felt like an ice bath to me, but I think it was just a cool bath.
So again, Sammy writes, it's a shame medicine is getting robotic. Many doctors put PRN as needed orders for Tylenol whenever a patient has a fever. So whatever the temp is above 100.4, the nurses automatically give it. Ugh.
No.
Yeah.
I wouldn't do it till at least probably 102.5, 103. You can correct me. But once you start getting up that high, then you probably want to keep it from going higher than that.
Vanity wants to know, what is it about the immune response that makes prior injured joints hurt again?
I wish I knew. I broke my wrist. I don't know. Show this over here. Yeah, it doesn't show up that much, but yeah, I broke both my wrists ice skating. Yeah, they hurt a little bit.
I think I remember that, right? It was not too long ago.
Yeah. I think it's just that you have some damage there, and so when you have new infections or new trauma, it probably promotes those same inflammatory responses that then trigger it more there. I wish we knew why it hurt when it got cold, because sometimes my wrist aches when it's cold. I don't have a great answer for that.
And back to 4 Los, this really interesting thing about mRNA vaccines is that their immunogenicity appears to be unaffected by TLRs other than 3.
Yep.
The PRRs responsible for their great immunogenicity are poorly defined.
That is right. There's probably components of the nanoparticle that are also triggering responses, maybe not through TLRs. But yeah, they have the mRNA and that will trigger TLRs. Although
all right, one more question. We're out of questions here. So here's one. Dagfin, is it possible that this joint pain is due to accumulation of circulating antigen antibody complexes?
Yeah, that's possible. They certainly cause joint problems. So if you have autoantigens or just antigen immune complexes that make it into the joint, they could cause a little irritation. Yeah.
All right, I think that will do it. It's 9:30. I told Cindy we'd probably go for an hour and a half.
That went fast.
It goes fast. We have great questions, as you see. So thanks, everybody, for coming. Let me thank our moderators. We had Les here tonight. We had Vanity Nutrition. We had Steph SF. We had Andrew over in New Zealand. Did I hit everyone? I think I did. Tom from Eugene, Oregon. Thank you all for moderating tonight. I know that you had to take out a number of people. I'm sorry about that, but that's the way it goes. There was one comment here that I missed. I wanted to hit. I'm not going to be able to find it. Anyway, folks, thanks for coming. We'll be back next Wednesday. That's January 24, 8 p.m. Eastern. We will figure out if it's me alone or if I come up with a guest at the last moment. But I want to thank you, Cindy, for joining us tonight. I hope you liked it, and maybe you'll come back at some point.
I would love to come back anytime. And anybody who's on here, go to the YouTube video and make comments about what you would like to see us talk about on immune as well. That would be great.
Yeah, as always, you can put comments and say, we'd like you to talk about this, and that way we get a heads up.
Yep.
All right, that's another exciting episode of Office Hours. You can't. Where else can you get this? Where you have professors who work on science just sitting here taking your questions. I think this is the coolest thing. I love live streams. They're a lot of fun, right, Cindy?
Yeah. This is definitely different than I thought it was going to be. It was great. I love all the comments that everybody had, and you guys had such interesting questions. Yeah.
We just have a last-minute contribution by Dr. Bro Shropshire. Thank you so much for your support of science communication. We really appreciate it. Okay, folks, that'll do it for another Office Hours. See you next week. In the meantime, please stay safe. Good night.
Good night.